All right, well, good morning. Last time we were looking at this amino acid sequence, and I had you go through and try to identify the positively charged amino acids versus the negatively charged ones. So what do you got? Which ones are plus? Pluses are K's and R's. So every time you see an R or a K, that's a plus charge. K is for lysine. Okay. All right, did I miss anything? There's another R. I think that's it. So that's plus what? 1, 2, 3, 4, 5, 6, 7, 8. And then minuses. We'll use red. That's E and D, these two E's, this one, this one, this one, and then the C terminus. So we've got overall 8 plus, and then 2, 4, 6, 7, 8 minus. Did anybody see anything there that I missed? Any pluses that I missed? Yeah, go ahead.
Is the N-terminus positively charged on the zero?
The N-terminus shouldn't be positively charged here. It normally would, but we've modified it with an acetyl group. It's an amide. The reason for that we can get into. There is a specific reason for it. I'm just presenting the molecule as it is when it's published. But yeah, so overall, if I didn't miss anything, overall the net charge is 0. And that's going to be important later on when we see what this molecule does. Okay, how about nonpolar residues? We'll highlight those in green. Okay. I don't know, red-green colorblind folks, that might be an issue. So let's do orange. All right, so here, as I told you, we're looking for things that are more nonpolar than, say, alanine. So a bigger nonpolar side chain than alanine. And we're also going to sort of exclude the aromatic amino acids. So M is definitely one of them. L, then a V and an L. Let's see, then another L here, another V, an L, and L, and a V. Okay. So here is this sequence, and if you look at it for a while, if you don't know the pattern is there, it's hard to see. But there is a pattern, and if we zoom in on just the first part of the sequence, one of the things you notice is that the nonpolar amino acid M is 3 amino acids away from the 3 spaces away from the nonpolar amino acid L. Okay? And then the next spacing between L and the next nonpolar amino acid is 4. 3 and then 4, and then there's another nonpolar amino acid at 3. And then if we go over here, we see a spacing of 4, and then 3, and then another spacing of 3 or 4, we're not sure. Okay? So there's this interesting pattern of nonpolar amino acids on every 3rd and then every 4th amino acid after that. And you'll see that the pattern isn't perfect. For example, if I draw, let's see, 1, 2, 3, 4. Did I mess up somewhere? Nope, I think I got it. Let me just confirm that I got the right sequence. Yeah, I'm missing an amino acid, friends, in this sequence. Can you tolerate that, or is that going to be totally unacceptable? Yeah, I mean, whatever. Sorry. So it turns out I wrote this sequence down last time in the notes. There was one missing amino acid, and it's another L, and then SKN. Okay? So I'm sorry about that. It doesn't change the overall charge. It changes the number of nonpolar residues. So now we're going to highlight both of these. There's a spacing of 3 to the first L. You see 1, 2, 3. And then after that first L, there's another 1, 2, 3, 4, and that would take you to this amino acid, but that breaks the pattern, right? It's polar rather than nonpolar. Asparagine is N. That's the side chain, if you remember, that looks like this. So that's nonpolar. Leucine is the side chain that looks like this. That's nonpolar. And then valine is the side chain that looks like that. Okay. So what I'm trying to point out is, starting with this nonpolar methionine, you've got 1, 2, 3, there's a nonpolar residue. Then 1, 2, 3, 4, there's another nonpolar. And then you go 1, 2, 3, there's a nonpolar one. Now there happens to be another one there, and that's fine. As I said, the pattern is sometimes messy, but if you count from this first L, it's 1, 2, 3, 4, and that's where we get a weird, we'll color it gray because it sort of breaks the pattern, a neutral but non-polar amino acid. Nevertheless, 3 spaces after that, 1, 2, 3, there's another nonpolar. Then 1, 2, 3, 4, there's a nonpolar. Then 1, 2, 3, and so on, 1, 2, 3, 4. So even though some nonpolar residues show up outside this sequence, like this one, which is sort of weird, and this one, which is sort of weird, there is a pattern of nonpolar residues that follows this 3 to 4 to 3 to 4 sequence. Yeah?
Sorry, you called N non-polar?
I'm sorry, if I said that, it was wrong. N is neutral, but polar. Whereas these two, leucine and valine, are both nonpolar. Okay. So this is the trouble with protein sequence, is there are patterns, but there's some wiggle room with whether or not you follow the pattern. So can you see the overall 3, 4, 3, 4 pattern, even though it's sort of broken by these 3 examples, and in particular this N at a place where you would have expected a nonpolar amino acid? People have noted this pattern. The 3 and the 4 add up to 7, and so they call this pattern a heptad, or 7 amino acid repeating sequence. They'll call the first amino acid in the sequence, they'll give it the letter A. So methionine is A, B, C, D, E, F, G, and then you start again with A. And the idea is every A and D position, you usually have a nonpolar amino acid, with the single exception of that asparagine that occupies an A position with the exception of this single asparagine that happens to occupy an A position. This is called a heptad repeat, and it's an important sequence pattern because it forms the basis of a class of molecules that you'll learn about in biochemistry that are sometimes called leucine zippers, and the other thing that they're called is coiled coils. All right, so to understand this, I don't necessarily want to move on until, because the punchline isn't going to mean anything if you have no idea what I'm talking about. So, questions you have on this pattern I'm showing you of nonpolar amino acids, spaced 3 and then 4 apart. Anything you want to? Okay, yeah.
Why is the nonpolar significant? Why do we care about that? Those types of amino acids as opposed to the other
Right, why are we focusing in on the nonpolar ones? My answer to that is we talked, I think, last time about what nonpolar amino acids do, and that is they tend to be sequestered from solvent, okay? So that's an important piece of the puzzle. Remember that in general, nonpolar amino acids, I shouldn't do this, but because they don't have feelings, but for our purposes, they don't like to be exposed to water because of the hydrophobic effect. So whatever happens to this protein, we're going to expect the nonpolar amino acids to not be exposed to water. Other questions? Yeah.
When you said coil, is that similar to like a supercoil?
Is that similar to a supercoil? The term coil refers to two, we're going to get there, I'll show what it means, but it refers to two alpha helices that self-associate and wrap around each other. So the one coil is the alpha helix, and then they coil around each other. Yeah. And we'll show you why in just a little bit. Other questions? All right, so before I show you what this sequence does, you need to understand a little bit more about the alpha helix than maybe you currently do. It's possible that you've heard of an alpha helix before, and it's possible you've heard it's sort of a spiral, and the peptide backbone spirals on itself. We want to focus in real briefly to look at what the atoms are doing in an alpha helix. So this is an image from a crystal structure, which is a three-dimensional structure of a peptide or a protein. X-ray crystallography is a technique where you take a peptide or a protein, you get it really concentrated, and if you do things just right, you can sometimes get it to form a crystal as it crashes out of solution. And then you can take that crystal and shoot X-rays at it, and the regular pattern in the crystal will diffract the X-rays. And interestingly enough, the diffraction pattern of the X-rays gives you sort of a blueprint for where the atoms are in that crystal. So you can get a three-dimensional picture of what a molecule looks like. And this is just the backbone atoms for a molecule that forms an alpha helix. So in this image, the oxygens are red, the nitrogens are blue, and the carbons are yellow. And you can see, all I'm showing you are the backbone atoms. The side chain atoms are totally omitted. So for example, starting with this nitrogen, here's an N, an alpha carbon, and a carbonyl carbon. That's one amino acid residue. We could maybe highlight that. No, we won't bother. In purple. Yeah, it won't show up very well. And then this is the next one. N, alpha, and C. Then this is the next one, N, alpha, and C. And then here's the one after that, N, alpha, and C. All right. So you can see, if we think about this, we'll call this amino acid in black the I position amino acid. The orange one we'll call I+1. The purple one we'll call I+2. And the green one we'll call I+3. And then over here in pink is the nitrogen and the alpha carbon of the I+4 amino acid. Understand that this language I+1, I+2, I+3, I+4 is all about just counting from an arbitrary reference point in a peptide, okay? One of the things you see is that it takes about 3 or 4 amino acids before the backbone spirals up and back around to sort of the same place, okay? So you can think about the helix as a cylinder. The peptide is wrapping itself around the cylinder. It's moving from the N-terminal side of the cylinder. This is the N-terminal side of the peptide, and this is the C-terminal side. And as the helix spirals around this cylinder from the N-terminus to the C-terminus, it circles back around the cylinder, but now it's further up the cylinder, okay? So by the time you get to the 4th amino acid, or the 3rd or 4th amino acid, you're sort of back in space to the same side of the cylinder, only you're further down, okay? There are people have characterized the alpha helix mathematically, and they figured out there's about 3.6 amino acids per turn of the helix. They've also figured out that there's, you move down the cylinder a certain amount, it's about 5 or 6 angstroms per turn of the helix. Another key feature of the alpha helix, the thing that really makes it so there's 3.5 amino acids per turn, it's 3.5 to 3.6, is the alpha helix has hydrogen bonds between the NH at the I+4 position and the CO at the I position. And you can see this all the way down. Here's what we call, this that I've just drawn here, we would call an I to I+4 hydrogen bond. And it's between the carbonyl oxygen of the I position residue, and then you count 1, 2, 3, 4, and then the amide nitrogen, the amide proton is going to be hydrogen bonding with that. You can see these all over the place. Here's one between this one that I'm highlighting here and this nitrogen. Here's another between this carbonyl oxygen and this nitrogen. So the helix has these regular hydrogen bonds all the way around, and they're always between the I position and the 4 amino acids down, okay? Now, this regular pattern at about 3.5 amino acids per turn, let's see what that would do. Let's actually start with our sequence. Remember, the sequence of our peptide, the key feature of the sequence is every 3 and then 4 residues after that, we have a nonpolar amino acid. So let's just start here. This will be our starting point. And let's say there's a nonpolar amino acid there. So let's go around, we'll count 1, 2, where do I want to start to illustrate this? Actually, I want to start with this one. Okay, let's say this one's the nonpolar amino acid. We'll count from there, 1 to 1, 2, and then 3. So according to our pattern, there would be a nonpolar amino acid here, and the space between those two would be 3 residues. Then we'll count up again from here, 1, 2, 3, 4. Here would be the other nonpolar amino acid, and the space between those two would be 4. What I'm trying to illustrate is that the spacing of nonpolar amino acids every 3 and then 4 puts nonpolar amino acids all on the same side of the helix, all the same side of the cylinder, okay? So if you were, schematically, if we were to draw an alpha helix, and if it had this repeating pattern of nonpolar residues every 3 and every 4, then what we would see is basically once every turn of the helix, we would have a nonpolar amino acid. And it would basically be along the same side of the cylinder. Okay? Does that sort of make sense? Because of that 3.5-ish on average, 2 turns of the helix at 3.5 turns per residues per turn, that means every 7 amino acids, you're coming back in space to exactly where you were previously, 2 turns previously on the helical cylinder. Okay, so now with that, let's imagine if this peptide formed an alpha helix, because of its pattern of 3, then 4, then 3, then 4, this heptad repeat where every A and D positions have nonpolar amino acids. If this peptide made an alpha helix, it would be what we call globally amphipathic. Amphi means both, and pathic means love. Amphipathic means something that has 2 different characters. It has one side of the helix that's nonpolar, and then we would see this other side of the helix is composed mostly of polar and charged residues. I'll try to illustrate this with it's called a helical wheel diagram that's intended to show you how the folding into an alpha helix displays side chains. Here is the same amino acid sequence we saw above. The nonpolar residues I'm at A and D positions. Excuse me, sneezing, I'm highlighting in orange. We're going to put that weird asparagine that sort of breaks the pattern in gray. And then for now, I'm not going to highlight the charged amino acids, though we'll do that again in a little bit. This is the view of this peptide in the crystal structure when it forms an alpha helix. We're only looking at amino acids in the first 7 residues. Here's the methionine at A. Here's the lysine at B. Here's the glutamine at C. Here's the leucine at D, the glutamate at E, the aspartate at F, and then the lysine at G. So you can see when this sequence forms an alpha helix, you notice the 2 nonpolar residues, methionine and leucine, are along the same face of the helix, on the same side of the helical cylinder. Excuse me, and you notice that the charged amino acids and the polar amino acids are all on the other side, okay? So, and if you turn this drawing into a schematic, which sort of circles around from A to B to C to D to E to F and G, this is called a helical wheel diagram, and it simply displays in sort of a neat way what you would see if you looked down the helix. You see the 2 sort of non-polar segments here. In general, they are mostly non-polar. There's a couple of things that break the pattern. One charged residue at the very end, which I'll just gray out, and one asparagine in the middle, which I'll gray out. Other than that, they're entirely non-polar. And then sort of over here, you have charged or polar water-soluble amino acids. Okay, now add in what you know about the hydrophobic effect, and you would predict that it would be bad for this molecule to be a helix on its own, because it's got one side that's basically almost entirely nonpolar. So, and when you expose nonpolar surface area to water, what happens is the water molecules immediately around the nonpolar surface area lose entropy, and that's unfavorable. They form a rigid ice-like cage. So to prevent that from happening, we're actually going to take 2 of these helices and associate their nonpolar faces together. That's going to take them out of water and into an internal environment. And what you get is this, which is called a coil-coil. So this is the molecule. The peptide happens to be named GCN4P1. P is for peptide. GCN4 is the name of a transcription factor from yeast that has this sequence in it. In any case, here is the one helix wrapped around the other. This is the N-terminal side to the C-terminal side. And then in this view, we're looking down the interface between the 2 helices from the N-terminus. Here's that first methionine residue and the second one. Here is the leucine residue on one helix packing against the leucine residue on another helix. You get this called a hydrophobic core of this coil-coil where the nonpolar amino acids are in the middle. Another interesting feature that's not captured by this image is that there are amino acids that flank the core, and frequently on one side of the helix there's a positively charged amino acid, and on another side of the helix there's a negatively charged amino acid, and they will interact across the helix across the interface between helices in what's called a salt bridge. Okay, a couple of things now about the charges. We noticed above that the net charge on this molecule is 0. That actually is important. What do you think might happen if the net charge on both peptides, on one peptide, It's important because the two peptides have to come together. What do you think would happen if the overall charge on this GCN4 peptide were like highly positive? What would happen as the two helices came together? You're gonna have some charge repulsion, right? And maybe it wouldn't matter at first, but as you build up increased positive charge, there'd be some repulsion that you'd have to overcome. That would destabilize the coil-coil structure. Similarly, if the net charge were totally negative, you'd get repulsion when you came together to form a homodimer. So one of the reasons this molecule can form a homodimer is not only that you have nonpolar residues at A and D positions, but also because the overall charge is neutral, it's not destabilizing to have the two helices come together. Okay, we're almost done on the subject of coil-coils. What I want to just point out is I want you to sort of think about the question. What if you wanted to design a coil-to-coil heterodimer? That means you want two peptides, two copies of this molecule, and you want them so that they don't form homodimers with themselves. You want a peptide and a B peptide that don't form an A homodimer and don't form a B homodimer. You want them to form an A-B heterodimer. Go ahead.
Would the coil be a tertiary structure?
Yeah, what is the coil-coil? Is it a tertiary structure? So just some terms that you've heard before. Secondary structure means local organization of the peptide into, say, alpha helices, beta sheets, or beta strands, actually, is the right way to talk about secondary structure, and turns. Tertiary structure refers to groupings of several secondary structural elements together. So, and that's usually in the same molecule. So this would be, say, a beta sheet where two beta strands link together. That's a tertiary structure. Quaternary structure is association of two different molecules. So this is intermolecular association. So the coil-coil is an example of a quaternary structure, unless one thing you can do to make it a tertiary structure would be if you somehow tied these two peptides together in the same molecule. So a coil-coil has secondary structure because it's an alpha helix, and then the fact that the two alpha helices in different molecules are associating together is what we call quaternary structure. Tertiary structure is exactly the same thing, only it's in the same molecule. Okay. Yeah, other questions? So the whole point of this is to illustrate the relationship between sequence and structure and ultimately function. In the yeast transcription factor from which this molecule is derived, you need this dimerization, you need the two peptides coming together so that a bunch of stuff on the other end of the molecule can actually reach around and bind DNA. Now that we know this rule about coil-coils, you should be able to look at a sequence, try to look at nonpolar residues in the sequence, see if they fit this pattern, and then draw some kind of conclusion about whether or not it could form a coil-coil. And again, remember, the pattern doesn't have to be perfect. You got to be a little bit forgiving. That asparagine that is at an A position where you would think it should be nonpolar actually plays an important role, but we don't really have time to talk about it today. But yeah, I do want you to consider the question, if you wanted to modify this sequence so that you could make two peptides that would fold together to form a coil-coil, but not fold themselves with each other. Do you know what I'm talking about when I say homodimer and heterodimer? We sort of getting that? Not really. Okay. So A homodimer is when two copies of the same peptide form a dimer together. A heterodimer is when two different molecules form a dimer together. So what I'm showing you here with this coil-coil, this is a homodimer. And if we call this peptide A, it's forming another peptide. It's forming a dimer with another peptide A. A heterodimer would be if you had A and then some other molecule B, and they fold together to form a dimer that involves two different molecules instead of two of the same. So yeah, could you redesign the GCN4 sequence based on something that we've talked about today so that you could get a heterodimer? And think about, the clue is to think about the fact that GCN4 itself, this molecule has a net charge of 0, which we talked about promoting homodimer formation. Okay, and people today now know you can look at amino acid sequences of proteins, and if they have this pattern, people will say that's probably a leucine zipper, that's probably a coil-coil, and you can actually test these predictions, and people know a ton now about coil-coils because of this rule, which is a demonstration of the relationship between sequence and structure and function. Okay, one more thing on an alpha helix, another example of sequence, structure, and function. So, and I'll post these notes. You don't have to scramble to copy down stuff. We're not about memorizing the sequence. There are a class of peptides in organisms across lots of animals called host defense peptides. Insects make these, people make these, frogs make these. Here is a sequence from frogs. It's called meganin, and this is from a Hebrew word for shield. And this is a peptide that's part of what we would call the innate immune system, meaning it's a peptide that's designed to kill bacteria. And if you look at the sequence, one of the things you notice is, and I've highlighted it for you here, the N-terminus is positively charged. And then I've noted the rest of the positively charged residues. K is lysine, that's plus. K, also another K, those are all plus. So we have 1, 2, 3, 4, 5 pluses. In terms of histidine might be, depending on pH, we're not including it because at pH 7 we're assuming it's mostly neutral. The only two negatively charged amino acids is this glutamate, E, and then this C-terminus. Overall, net charge of, let's see, plus 5 minus 2 plus 3. Okay, so this is a cationic peptide because its net charge is plus 3. Now, if this peptide folded into an alpha helix, shown here is a slightly different helical wheel diagram. The one I showed you earlier is for molecules that are in peptides that form a coil-coil where there's about 3.5 amino acids per turn. This helical wheel diagram is for peptides that form just normal alpha helices where there's 3.6 residues per turn. Doesn't seem like it makes a small difference, a large difference, but the helical wheel diagram is different. I wouldn't have expected you to come up with this on your own. If I were to give you one of these and you can follow the lines, you could sort of map out going from 5 to 6 to 7 to 8 to 9 to 10 and so on. If you map out this sequence onto this helical wheel, I've got all the non-polar residues, including the aromatic ones here in green. And again, you start to see this globally amphipathic helix where you've got one face, which we're now using the color green for, that is mostly nonpolar, and then you've got another face that's mostly polar and is comfortable in water. Okay? So, similar sort of thing that we saw with that coil-coil peptide. The sequence pattern is not the same. It doesn't keep the same heptad repeat pattern, but overall, if you fold it into a helix, it would still have this globally amphipathic structure where one side was mostly nonpolar and the other side was mostly water soluble. Now, how does this kill bacteria? What's the point? You need to know a little bit about bacterial membranes to understand the point. So behold the lipid bilayer. Yes, behold, the lipid bilayerinator, Doofenshmirtz. I think of him every time I hear the word behold. So, yeah, you should always say the word behold. When you're reading the scriptures, you should always say the word behold, like Heinz Doofenshmirtz. My father-in-law actually has a brother Roger, and I'm trying to get him every time he talks about his brother Roger to say, my brother Roger! But he tried it once. It wasn't as good as me, but we're working on it. Okay, so the thing you need to know about bacterial membranes is they're phospholipids, and we will learn about these in more detail in a subsequent chapter. Those are mostly negatively charged. So I don't know why I chose to use yellow. Yellow's for polar head group, but these are mostly negatively charged. Now, the lipid bilayer separates what's inside the cell from what's outside the cell. Outside the cell is water, inside the cell is water. The other thing you need to know about bacteria is they don't have chromosomes like you and me. Their DNA is just packed into and stuffed into the cell. And it's packed pretty tightly. If you could manage to burst the membrane or open the membrane up, create little pores in the membrane, the contents of the bacteria are going to spill out into the outside and the bacterium dies. Remember, the membrane is really the only thing that's separating what's you from what's not you. And so if you can break that barrier for the bacteria, then they can't continue to replicate and they die. So how it turns out that this peptide will kill bacteria, and the way it does this is related to its globally amphipathic character. So this is a short sequence, and it's generally unstructured on its own, but when it gets in the presence of a lipid bilayer, it folds into an alpha helix so that it can have a nonpolar face. Then that nonpolar face starts to intercalate into the interior, whereas the positively charged face starts to associate with the negatively charged head groups. All right, so let me just walk you through that again. Imagine this sequence is just extended, floating on its own, but it gets near the negatively charged lipid bilayer. That provides a driving force for it to rearrange itself so that all that its positively charged amino acids are on one side of the helix over here. It folds into a helix in order to have all its pluses on the same side. Then that positively charged side can start interacting with the negatively charged face of the lipid bilayer, but it's also got a nonpolar side. And so if we zoom in to this cartoon and think about these helices as just cylinders. The color is unfortunate here. I should have chosen a different color scheme. But you can see the plus side, the positively charged side of the helix interacting with negatively charged head groups of the lipid bilayer. So I'm just going to put minuses here. And then over here, which I'll now use gray, is the nonpolar face of the peptide, which is interacting with the nonpolar interior of the lipid bilayer. This is a result of a simulation showing this kind of thing happening, where these start to poke holes in the lipid bilayer, and then the guts of the bacteria sort of spill out, and the bacterium dies. Now, why doesn't this do this to your cells? The answer is your cells eukaryotic cells generally don't have a net negative charge. They have a neutral or sometimes net positive charge. So there's some selectivity. Your body can make peptides like this and it will kill bacteria, but not you. The one that your body makes is called L27. Here's the sequence, and you don't need to write that down. But if you were to fold it into an alpha helix, you would see that LL37 also has a positively charged face and a negatively charged face. We can just go through real quickly and identify the positively charged residues. We'll color those blue. That's not blue, that's green. So there's a K, another K, another K, another K. I missed the R. There's another R, R, K, R, R. Did I miss anything else? And then there's a few negatively charged residues, but not that many. There's E, E, there's a D, there's another E. Overall, if you added this up, highly positively charged with some balancing negative charges. That's going to provide the selectivity for the bacteria versus eukaryotic cells. And then let's go gray for the nonpolar residues. Anything that's more nonpolar, say, than alanine. And you can see there's a ton of them. And if we folded this into an alpha helix, you would find these nonpolar residues in general on one face, the charged residues on the other. And again, this is going to disrupt bacterial membranes. Okay, questions? Another relationship between sequence, structure, and function. All right, I think, am I going to tell you the beta sheet story today, or are we going to save that for another time? I think you can't decide. Some of you are like, yes, please, and some of you are like, no, no more, let's just be done. All right, I'm afraid I'm going to get that wrong. Okay. All right, now we'll talk about this briefly. I hope you can see that we're doing something different than we've done in the class before. In the class, in earlier lectures of this class, earlier times we were together, you were scrambling down to capture things, and if you didn't write it down, you thought you wouldn't get it, and there's mechanisms you needed to know. Now we're looking at molecules that are too big to draw. We're looking at slides that are too large for you to copy down. The notes will be available to you, and I'm afraid some of you are concerned, do I need to memorize all of this? I feel like there is some memorization, because today, if you didn't know that K was positively charged, you're probably feeling a little bit lost. And if you didn't know that leucine and valine were nonpolar, you're probably feeling a little bit lost. But the point is to use stuff you know about properties of amino acids to understand structure and function, mostly at a qualitative level. If I showed you a sequence and I said, could this form a coil-coil, you should be able to answer that question by looking for where the nonpolar residues are. If I showed you a sequence and said, is this thing going to form, if it is a coil-coil, could it form a homodimer? You should be able to answer that question based on the overall positive or negative charge. If I asked you to explain why coil-coils self-associate, you should be able to answer that question with ideas about the hydrophobic effect and how that works. The story I want to tell you now, and again, you can learn to draw a beta sheet like I've drawn here. I could have taken the time to draw it with you, and that would have been good, but I think if we had done that, we might have missed the point. The point is not can you draw it, but the point is to understand sort of what a beta sheet is and to sort of draw some conclusions. So in a beta sheet, the backbone is in an extended conformation. So here's a nitrogen, alpha carbon, carbon, here's another, and you see it adopts this sort of regular zigzag pattern. Just the way you would draw a peptide even if you didn't intend for it to have any secondary structure. This is called a beta strand, and you can see that in a beta strand, the side chains, the R groups, are alternating on different sides of the strand, right? That's just because of the way the zigzag works. There's a dash side chain here, a wedge side chain here, dash, and then wedge. This particular kind of beta sheet that I'm showing you is called an anti-parallel beta hairpin. And in that anti-parallel beta hairpin, there's a 2 amino acid turn, and then we start again. So you can see we're drawing the N here represents the N terminus of this strand towards the C terminus. N to C, then we have this 2 amino acid turn, and then we've got another strand, only now this is the N terminal side, and it goes N alpha C, N alpha C, N alpha C in this zigzag pattern. This is why we call it an anti-parallel beta hairpin, because if the 2 strands are arranged opposite to each other, N to C here, and then N to C here, okay? So this is the simplest beta sheet there is. and you can see the hydrogen bonds between strands. But they're not between every amino acid in the strand. That's kind of interesting. The orange amino acids here, you can see the carbonyl oxygen on one strand is hydrogen bound to the opposite NH on the other strand. And within that same amino acid, the NH of this amino acid, which I've labeled the N in blue here, is hydrogen bound to this carbonyl. We call that a hydrogen bound pair. Now, because of the zigzag pattern, you can see the next amino acid, the carbonyl, NH, and CO are oriented on opposite sides of the strand. They're pointing away from each other. So that's a non-hydrogen bound pair. And in this anti-parallel beta sheet, you've got alternating non-hydrogen bound, hydrogen bound, non-hydrogen bound, hydrogen bound. The other interesting thing here is that the amino acids in the hydrogen bound pair, the side chains are pointed out, sort of away from the space between the two strands, whereas the amino acid side chains for non-hydrogen bound pairs are oriented towards each other. You might expect there to be some non-covalent interactions between these side chains in non-hydrogen bound pairs, whereas there wouldn't necessarily be between the side chains in hydrogen bound pairs. The other interesting thing about the beta hairpin is that these two amino acids have to kink around quite quickly, and they sometimes have to adopt weird dihedral angles to do so. For example, it's been observed that in most beta turns, so they call these four amino acids the hydrogen bound pair immediately preceding the turn, and then these two in the middle that connect the two sides, they call that a beta turn. And they call the first amino acid in the hydrogen bound pair I, then I plus 1, then I plus 2, then I plus 3. And these two amino acids sort of that make the turn have to kink back on each other, and these are the I plus 1 and I plus 2 amino acids. If you measure their phi-psi angles, they tend to be, in beta hairpins, in some, they tend to be different at least than typical. At I plus 1, they prefer to be minus 60 and minus 30 for phi and psi respectively. That's close to what you would expect for an alpha helix, so there's actually a lot of amino acids that work there. But the I plus 2 position is weird. That's plus 90. Now, if you remember what we talked about last time about the amino acids, there's not very many amino acids that like to adopt dihedral angles in the plus 90 region of phi. Okay? So you would say alanine no good, valine no good, leucine no good, isoleucine no good. Keep looking for them. Nope, still nothing at, okay, what about histidine? Well, that has sort of plus 60 and plus 40, but not really plus 90. Keep looking. What about aspartate? Again, not quite. Glycine, right on. Yep. So glycine, because it's more flexible, can adopt this dihedral angle. It's right there. And so what you tend to see for these beta hairpins is it's really, really common to have glycine at this I plus 2 position. Yeah.
Is that also because glycine's like the flexible one?
Yeah. Yeah, the lack of the side chain on glycine allows it to adopt this dihedral angle that's way over here where there would normally be steric clashes. And people have used this. People have used this information to design beta hairpins that are more stable than you would expect just by putting glycine at this particular position. All right, so and I need to look at this because, I'm actually wrong on the sign on that. The typical dihedral angles at the I plus 1 position are also positive. So we'll talk about that just briefly at the beginning of next time and choose a good amino acid for that position. All right, see you Monday.